All right, cool.
All right.
Yeah, so I just wanted to quickly introduce as well Clemens. Clemens is visiting us for there's a major conferences going on in Montreal starting Saturday, I think, OHBM. So it gathers a lot of people in neuroimaging in particular. And they do a lot of medical imaging, a lot of brain analysis, a lot of all related topics. And Clemens is coming from a very good lab in Germany. So they do heavily focused on machine learning, the deep MI lab as well. That's the cooling, I think, led by Martin Reuter as well. Martin still is sorry, doing a lot of shape analysis, a lot of machine learning today related to how to understand the brain, basically. And Clemens wanted to show some latest work that will be presented, if I understood, as well at HDM. So yes. Thanks, Clemens, for visiting us. It's a privilege to have you.
Thank you for the invitation. Thank you so much for the kind introduction. As you're introduced, I'm a PhD student in Martin Reuter's lab in Bonn, in Germany, and I'm in my third year now. And my PhD is basically on the robustness of the brain segmentation tools. So I'm looking at different ways these toolings can fail, like free surfer or fast surfer toolboxes. And I want to introduce some of those failure modes to you today, and especially focused on motion, which is what I'm presenting also at the OGPN. So this is the campus and the Institute for Neurodegenerative Diseases. So it's all focused on the brain. And this is where I work with my colleagues. And we also have an external site there, which is the Rhineland Study Centers. So these are basically research centers where people only go for the Rhineland study. And there's a multi-stage process where people get blood collected, MRI scans are taken, gait analysis is done, and many, many more tests, questionnaires, so on and so forth, where we collect a lot of data. And I'm fortunate to collaborate with them and to also apply our methods that we have for this brain segmentation on the Rhineland study.
Is Rhineland for Alzheimer or neurodegenerative disease or just general?
It's neurodegeneration. So basically aligned with the Institute's mission on dementia, Alzheimer's. The people that are recruited is everyone from age 30 and above from this area of Bonn. So it's a prospective study. And people are re-invited after, I think, second round is after 3 years, maybe a bit longer, 3 to 5 years. And then we also have longitudinal data to analyze and then see the development in the early stages of these diseases. And now there are over 10,000 participants that went through all this course here and had many markers taken. And the one I'm most interested in is here, which is the MRI room, where these participants lie down and get imaged for 1 hour approximately, and we have all kinds of sequences there, and that's where we are very interested in applying our methods. But of course, not only there, here's a work from my colleague Leonie Henschel, who did, as an example here, disease group analysis. This is with the fast surfer toolbox. So basically the deep learning based version of free surfer, which is running faster, but can also perform these disease effects analyses. So I think most of you are probably familiar with this, but we can see here control versus dementia, that the brain structure is different, but we can also see that we have different cortical thicknesses, and we can see the volumes of the structures through the deep learning based segmentation. And this is what we want to do in the Rhineland study, in the OASIS study is shown here, but also in all kinds of other studies. So really the idea is to take the methods into the real world and find some correlations and in the end understand the disease better. Now, what usually happens when we take it to the real world is the real world kind of strikes back. We want to do these cool things and we have our methods and data sets, but things happen. For example, the things I'm very interested in right now are unexpected structures like lesions or brain tumors, which the methods are usually not trained on. Deep learning based methods have never seen these cases, so they might give unexpected results. And also, for example, free surfer commonly fails on large lesions, and this is an issue. And motion artifacts are a bit different in nature, where, of course, the lesions change the actual structure of the brain, But motion artifacts are basically measurement errors. When people move in the scanner, we get unexpected measurements that are not true. And these can also cause failures and biases. And what is important here, and I will show a bit later, is that these biases can also be very subtle. So we need to come at this problem from different angles. And of course, there are many more cases, which I think can basically be divided in these structural changes that are unexpected and artifacts, which are wrong measurements, right? And again, I think many of you are familiar, but failure cases might look like this, where we can have nice images that work well with our pipelines, but then we can have, for example, a glioblastoma here, where the brain is then deformed and the free surfer-based registration can fail. But also our deep learning tools have never seen these kind of cases and therefore give unexpected results. What we do see though with, for example, 2-point-D approaches we have with fast surfer is that usually if you do slice-wise segmentation, some areas can still work well, but for example, for these large glioblastomas, there's few slices that are not affected, and therefore we need to address this. And the other case here is a very strong case of motion artifacts, where if participants move a lot during the scan, the segmentation can obviously fail because we cannot detect any structure, but there are also subtle biases, and that's what I mainly want to focus on today. So these motion artifacts and the work I've done on that. And what it boils down to is that we want to also discover these subtle differences, right? So here is an MRI of myself. One time I moved a lot, and the other times I moved a little bit less. And I think everyone can tell that the segmentation task will be very difficult in this case, where basically the edges are blurred. Probably on the TV is not very clear, but to make out any borders here is already tricky. And here we see some ringing artifacts, but we can make out the borders of the cortex quite well. And in these two cases, at least I cannot really see the difference when I move more or when I move less. And this is something that we want to address because we know that the biases still persist in these very low motion cases. And we basically want to find out how much motion occurred, right? And which of these images is actually the best quality one. And so to satisfy your curiosity is the one on the bottom left. That's the best quality image. But yeah, I think it is very hard to see with just by looking at the image. And this is important because of the biases that, as I now mentioned multiple times, persist. Martin Reuter, I had already shown this 2015, where here for the SBM toolbox, we see gray matter volume on the y-axis and the motion with the displacement on the x-axis. And so here's the same participant scanned multiple times. So one line is one participant. And what we see is that with more head motion, the gray matter volume seems to decrease. So basically what happens is if people move more, it looks like their brain gets thinner, the cortex gets thinner, they have less gray matter, which could be a sign of disease. But in fact, here is just an increased movement. And this is, of course, a bias that we want to avoid in our data. And this is true for the SBM toolbox, but also for FreeSurfer, which we see here. And now what people commonly do, for example, in the random study or in other studies, is that we just look at the image, right? Just like we did now. We had the 4 images and we could clearly see, well, 1 or 2 of them, they were motion affected. So we don't want to include those images in our analysis. But when we do this, we see that these biases still persist. And we still have this linear correlation between the motion and the gray matter volume. So therefore, we actually know that the manual checks alone are not sufficient, and we need some more sophisticated methods to actually detect and measure when does motion occur, how much does it occur. And we want to develop some automated tools for that. So what is the mission of this, and why did we choose to first look at the quantification of motion instead of, for example, going directly for motion correction? Well, I think there's a few open questions. First, it's not 100% clear which populations actually move a lot. So when is motion a big problem? How big is the problem? We know that, for example, in autism or in advanced dementia, there can be high motion, but the studies on this are rather thin, and sometimes they're also not directly comparable because the ways of measuring motion are different, and then we cannot really say, okay, this population usually moves more than that one, or to make these kind of conclusions is quite tricky. And then, of course, the applied researchers, they want to know how much is my analysis actually effective, how big are the biases I can expect, and what happens really downstream in also a larger context. And then I think another later question is then, when should we introduce these correction algorithms, right? Should we introduce them already for these very low motion cases where we know biases persist? Or should we maybe only use them when we have strong motion artifacts to avoid any biases introduced by the correction? And to disentangle these effects, we first need to have an accurate quantification, I think. And finally, by quantifying motion, we could then correct motion in statistical analysis, where we just use motion as a confounder and say, well, if people move more, we have to account for this in the calculation of our correlations and get these adjusted p-values. And so that's basically the goal of this project. We want to quantify motion during the MRI image acquisition. We want to know, when people get scanned and the brain scan is taken, how much did someone move? And so, yeah, this is the wish list, right? We have our images here, structural images, and we want some kind of score. I call this motion score. And what we want from this is we want it to be the actual motion, right? Not perceived quality. We don't want to have the label 1 or 0. Looks like it's bad, or maybe it's good. But we want to have some actual ground truth or measured motion. And we want to best aggregate this into single scores, because of course motion is a complex signal. It's a 3D signal over time, so 4D, and therefore we want to kind of aggregate this so we can actually use it in some statistical analysis and not have these thousands of values. And so of course we are in 2023, right? And we want to do deep learning. So we just take our images and we say, well, we want to predict some motion. But of course, we don't have any ground truth for this, right? We don't know how much people move when we get the images. We just have these qualities we can look at. So what we did is we say, well, we want to actually measure this. And so before we can do our learning things, we actually have our measurements here with an optical camera that is installed in the scanner. And this camera is a commercial motion tracking camera that can record these kind of images of the face with over 30 frames. So it's mounted in the scanner. I will show the picture in the next slide. And we can basically record people while they're being scanned, and from there estimate the motion, basically measure it. And this is then applied in the Ryan study, where we now have a subset of 500 participants for this work with all kinds of acquisitions available to us. Now, of course, it's never that easy, right? We just install the camera and it just works. We have to look a little bit more into how this is acquired. First, you can see here that the camera sits on the head coil and basically has a tiny field of view, right? So it's kind of on the top left of your face, and it records this general area, as you saw in the previous image. And then from the camera, this gets transferred to the system of the company, which then generates some point cloud data that we can see, and the study assistants can also see during the scan, which is quite nice. They can have an idea how the participant looks and what they're doing. But it also takes some setup, of course, which is contrary to maybe other methods, rather fast though. You basically just have to adjust this optical camera on a rail and fix it so it points at the participant's face, and then start the tracking. Now the tracking is done on then this kind of images. This is once again me in the scanner. And yeah, you can see here is I'm blinking a bit, and here's a little bit of my hair in the top. Here, these are parts of the head coil, and I'm lying quite still, right? It doesn't look like a lot of movement. But I think from gauging the actual head motion from this, we can already tell that, well, this part is probably not very useful. Here, this head curl is always stationary. These are static parts. So really, we're interested in these kind of more rigid areas here along the nose and the cheekbones. And for the registration, this is what's commonly done. So we remove first all these areas to get kind of more rigid parts here. And we're interested in understanding this, of course, because we're not 100% confident in the measurement method itself. We saw that there's some challenges there. For example, in this video where I move a bit more, we see all kinds of artifacts and basically a more challenging registration task where you can see that there are some wave-like artifacts here on the face, which is common for these kind of cameras. They are infrared time-of-flight cameras. So when motion occurs, there will be some distortions in the recordings. And also there's a limited field of view where if people move a lot, well, you see a different part of the face than you saw in initial images, for example. And so, of course, the task is not trivial if we just try to match point by points, like with a simple iterative closest point registration. That can not work, but we need to think about how we can address these challenges of noise, artifacts, and the partial registration. And for this, we proposed the reweighted ITP algorithm, which is previously introduced by Bergström et al. And it's the traditional iterative closest point method, but with an additional reweighting. So this is this term here, where you can see the points that are shown in red are assigned weights close to 0, which are then not considered for the registration, while the points in green are considered for the registration. And we introduce a second term, which is a regularizer that basically prevents unrealistic movements at the back of the head, because we only record the front, and then we dampen some effects that might occur at the back of the head, because in the literature, it's also known that people usually move around the contact point in the scanner. Like when people lie down, they move around the point where they are lying. So we try to also input this knowledge in our modeling. And with that, we can then get some of these motion traces. We see, well, here are the different acquisitions, functional MRI, T1, T2, flare, so on and so forth. So we have 1 hour, and we can see, well, here in the break, for example, where there's no acquisition, someone moved a lot. They were maybe talking or readjusting their position. And so we already get some nice data and some nice input. But finally, we want to get to our motion scores. And for this, we use Jenkinson's RMS deviation. So that's basically a weighting of the translation and the rotation, right? When you have the movements and people move in 3D, what we want to know is how much of the brain area is basically moving. So we have an integral over the sphere, and we average basically what we see here. We average this per acquisition, and then we get our score. So it's basically independent of the acquisition. We just get an average. How much did someone move when the image was acquired, right? And with that, we have our method that we developed ourselves. And we also have now the vendor supplied method, which we also want to test. But the question, of course, is we have now these 2 parts. First, the registration, which is competing with the vendor supplied method, and also our aggregation, which is novel. And once again, we have the problem, we don't have any ground truth, right? So now, well, we wanted to create a ground truth, but now we have suddenly a second method, which once again, we don't have the ground truth for. And in this case, it's very difficult to acquire because we already have accurate measurements, but of course, we don't want to buy more cameras and more devices to acquire the motion. And it's tricky to actually get trustworthy measurements, because that's basically what our method is trying to create, right? So what we did here is we decided to go for indirect validation. And for this, basically, we take other measurements that we don't consider necessarily to be ground truth, but we consider them to be close enough. And we consider them to have some properties that are better than our method, while other properties might not be as good. And so we think if we take multiple of these comparisons and we get consistently better results than the competing method, we can trust that this validation actually confirms the performance. And the first of these measurements is maybe intuitive, where if you're familiar with functional MRI, you know that there's multiple frames acquired. In the run study, we have 1,024 frames. And so we can use existing tools to estimate the motion from there and basically compare this. It's really nice because we get a very different measurements, right? We have the full brain, the full head, and we don't have this area of the face in the front, which can give us possibly very different estimates if our registration is not working very well. But the difficult and the tricky part is that it's rather low frequency, so our method operates much faster than the fMRI based estimates can be generated. And because the fMRI based estimates are also rather slow, they are themselves sometimes affected by these motion artifacts, right? Because they're also single frames, basically single MRI images. And when a fast movement occurs while this image is acquired, we then get an artifacted image that maybe doesn't work so well for the registration. So of course, it's not a perfect ground truth, but we can compare with this. And then we also wanted to get another more high frequency signal. And for this, we take a respiration sensor that we have in the running study. So basically what happens if people breathe in and breathe out, this respiration sensor just registers how the chest is moving. And this doesn't give us any real head motion estimates, right? But it gives us a very high frequency signal that can partially explain this head motion. So we know that we can see the breathing also in the head motion. And we want to quantify how well our method can capture this. So it's basically checking the sensitivity of our method compared to others. And finally, we want to test the whole pipeline. So we check how the motion scores that we get in the end are actually related with the image quality. Now, of course, these quality estimates, they can capture other artifacts, right? They're not exclusive to motion. And they're also specific to the acquisition. So we don't really know which measure captures motion the best here. But when we take a bunch of these scores and we compare, we hope that we can also get some meaningful insight whether our registration algorithm actually captures something that can in the end be seen in the image, right? Because that's still an important part. So without further ado, I will show you the results. First, the difference to the fMRI. Here, once again, we have our data and the fMRI data. Here the fMRI is not sped up like in the previous slide. And again, there's these challenges that we overcame where we basically average our estimates during the time one fMRI frame was acquired. And then we also have to map both of them to the same reference space. We register to the T1 image. And there's also another step which one has to be careful of, where we don't want to do, basically we want to avoid the bias of this registration. So if you have more questions on this, feel free to ask. But basically the idea is that we average over all possible reference points to not make the comparison biased. So we took quite some care here that we got a fair comparison between the methods. And what we see then is that we take no registration as a baseline, where basically zero motion is estimated. In tracksuit is the vendor supplied method, and our method is here in green. And MTD is the motion trace difference, which is the measure introduced by us. So the difference between the fMRI-based estimates and the no registration. So zero is larger than our method, which is expected, right? Because we expect that our method, the motion that we measure, is similar to the one in fMRI, so we expect low scores. But it seems like the vendor-supplied method is more different to the fMRI than just not estimating anything at all, which was quite surprising to us. And we suspect that this is because we have quite low motion in the running study. I mean, people are usually healthy and don't move a lot. And therefore, any noise that might be introduced by the vendor-supplied method might actually hurt the registration so much that it seems to be a bit worse than just assuming nothing at all, at least when compared to these fMRI estimates. Now, for the second analysis, we compare the chest movement here measured by the registration signal with, once again, our method and the vendor-supplied method. And once again, you can see here quite noisier estimates. And especially in this area, for example, you can see while our method tends to loosely follow the rhythm of the respiration here, but in the vendor-supplied method, this information seems to get a little bit lost. And this is exactly what we also see when we compare the mutual information of the registration, of the respiration signal with the motion information that we get from the registration. And so here we have random motion as a baseline, and the mutual information with the respiration is higher for the vendor-supplied method, but for our method, it's even much higher than that. So we really see a nice correlation between the breathing of people and how their head moves, which makes us confident that we get very sensitive measurements that are also accurate in this high sampling frequency that we have. So basically in the previous comparison, we averaged a bunch of signals, which maybe reduces the noise a bit. And here we might be able to see really if one of the methods is very noisy. And finally, we compare with the image quality metrics. I think this is rather straightforward. We take a bunch of metrics from MRI-QC, which are basically designed heuristics where we see, okay, how much noise is in the image, how sharp are the edges, so on and so forth, how much intensity is in the background. And we can see that the motion scores derived with our registration method have much higher correlation than the motion scores derived with the vendor-supplied method. The scoring here is the same, but the underlying registration is different, and we can see higher correlations. And I mean, you will notice the correlation coefficients here are not super high, but when you consider that we do not know anything about the acquisition in this part, and we just average the motion, I think it's nice that we get some low p-values and we get some significant correlations where we see, well, actually, even when we don't consider the acquisition and how everything interacts in the scanner, just by averaging the motion, we already see that we can get some information about the resulting image quality. And so that's our measurement method, right? And the people in the scan center in the RUN study, they are already happy. And we can get these motion estimates. We can use it to correct in the downstream analysis tasks. But what about the other scan center? Right, there's more than one. There's a second one, and we only have the camera in one center. And so this is where we get back to the deep learning. And then we want to ask, well, can we actually get these measured scores then from the images directly? And with the quality correlation, we already showed that there's some information, but now we want to see how far we can go, right? So we take the structural images and a fully convolutional network that is inspired by brain age prediction, which is another regression task in medical imaging, right? And we use a fully convolutional 3D network, as I said, bins of soft labels and the Kullback LiDAR loss to train this. So basically, we predict the probability distribution instead of just the scalar scores. And this seems to help a lot in this case. And what we see is that when we compare this tool to other architectures, that our architecture outperforms others like DenseNet, the brain age prediction network, or MIQA CNN, which is a CNN developed for the quality assessment based on annual raters labels. So it's basically classified. And we even see that the MIQA CNN fails to generalize. So the R² score here shown in orange is worse than, or is less than 0, which means on the test set, it's a worse prediction than predicting the average of the test set. And we also show the Spearman R, so the correlation, and we show this for the architectures and for some other tools. Now, these tools, they are predicting the perceived motion. Our tool is actually the first method that predicts actual measured motion, right? So this is a comparison with the rater-based motion, where other networks or heuristics are validated and trained on basically these yes or no labels that people gave for low quality, very low quality, good quality. And what we see are also lower correlations. And interestingly, for the MIQA toolbox, which is trained on these manual labels, we get a motion score of 0 for all of our images. So basically, all of our images are labeled with no motion at all, which I think is consistent with rated perception, actually, that the images seem to be of quite high quality. And we can do this not only for T1 weighted images, but also for other images, right? Just out of the box, we can retrain. And for all of the images that we acquire in this session, we get similar results, which is nice. So we can extend this, and we can, apply our method in all of the scan centers. However, I don't think this can be applied to very different settings. So for other scanners, for example, when the acquisition is very different, you will probably need to retrain the method as well, because the correlation or the relationship between motion and the image quality is very dependent on the acquisition and on the scanner and the way this interacts. But what I think is interesting is that we have now this head motion quantified, and we can, of course, use this in our statistical models, which is something we are currently working on and implementing this in the Rhine study, and seeing if we can explain some effects there. But we can also ask more questions about the causes of head motion, right? Which is something I've introduced in the beginning, where we can understand better how these biases are created. Let me quickly check time. Oh yes, I guess I will finish, so we maybe still have some time for questions. And what we see is basically that age and BMI are highly correlated with motion, and they are also correlated with disease, right? So these are two effects that are related, and we need to measure and quantify the motion to disentangle this. We see that motion increases throughout the scan session. So after the 1 hour in the scanner, we see up to 36% increase, and this increase is even higher with increased BMI and age. But also good news is that if you have, for example, motion during an initial fMRI sequence, this can be a strong predictor for later motion. So if you get any kind of measurements during your scan session, you might be able to use it to correct for a motion that is occurring even later. And we also have some of these manual quality ratings, and we saw that our methods are working in this area where motion is basically not visible, where we have no labels with strong motion artifacts and only 2.2% with any visible artifacts. Yeah, with that, I would like to conclude. Of course, there's more in the works. I mean, we would like to really apply this and figure out how the biases are created, how large they are, all the things I introduced in the beginning. But yeah, with that, I would like to thank my lab, Martin Reuter, my PI, and my colleagues, PhD students, postdoc, and of course the Rhine study as well, where there are many people working on this data acquisition, and they've been great to collaborate with. Yeah, I'm open for any questions. Thank you for the attention. Yeah.
All right. Well, first, I want to thank you for this amazing talk.
Thank you.
It's just very, very interesting. I had a question regarding how you aggregate the motion, and I was wondering whether some types of motion lead to stronger artifacts, and whether this aggregation is kind of like using this information.
I think yes. Basically, the answer is yes. So in this work, we focused on this average motion score, right? That's, I think that we just take, during one acquisition, we just say, well, the average motion, no matter when, how this occurs, so on and so forth. And this is for sure lost when we, for example, do the prediction that there's, of course, different types of motion, where respiratory motion is just this very periodic thing. But when someone, for example, coughs, well, maybe they have like one big motion and then they end up in a different position, so on and so forth. So I think there's arbitrarily many modes of this kind of thing. And of course, it depends on the timing as well. We know that motion in the middle of the acquisition is usually more detrimental because that's where the center of the K-space is acquired, right? So the large intensity changes will be caused by motion there. And then I think this is an interesting avenue for further things, actually. So the work I showed on the prediction, I present this as a initial work in ISPE this year. And I hope to follow up on this a bit more to basically take out these details and see maybe we don't only have one, maybe we have 6 scores or something, right? We could have probably hundreds of thousands of values that we give in from the measurements that we have. But in the end, it would be interesting to see what is actually an optimum or what are good descriptors for that. Yeah, very good question. Thank you.
I have another question, but I'll just want to make sure everybody has the chance to ask. All right, so I'll just ask my follow-up. So, I mean, this is just not like brainstorming, but assuming that there's a strong correlation between image quality and motion, and you can sort of predict the motion, could you use this as a way to modify the image in such a way that it improves the quality? So, for example, let's say that you could use this as a loss, and you want to minimize. You want to modify your image in such a way that the perceived or predicted motion is lower.
Yeah, I think this, so this is not done with our method, but other methods are already having this kind of losses, I think. So they have discriminators where they say, well, we want to improve the image quality. And I mean, it would be interesting to see because again, like this is the first one that's based on the actual motion, but here we don't claim to be highly correlated with image quality, right? So this, I think this will be the step even after, then, okay, once we get this relationship between the motion and the quality, right? Then we can say, well, now we know what the quality should be. Yeah. And then I think the ultimate question is, do we go from the retrospective point where we say, okay, we just take the image and then we try to optimize the image, or do we just modify the K-space or, right? Because I think this is always the trade-off. When you get good measurements, you can always do the prospective correction, but it also has its shortcomings, right? So,
and you mentioned that sometimes doing the correction introduces some bias or some artifacts. And what kind of artifacts would you have if you attempt to do a motion correction and your estimate of motion is incorrect?
I'm not super familiar. I know that basically if you correct the K-space and you interpolate there, you will get some kind of blurriness, right? Or you will have, especially in deep learning cases, also you get some modifications that are not ensured to be actually natural and maybe could introduce something, but I'm not familiar. I'm not even sure if there's any biases really shown. So this might be going a bit too far to say, well, this introduces biases, but it might introduce an unnecessary processing step that you don't know how it's going to behave. So I think the answer is, I don't know. We don't really know yet. Yeah.
Thanks.
Because I think one of the core problems with these correction methods is they're also validated on vastly different populations. There's some papers who validate their motion correction on people that basically lie still and have been in the scanner many, many times. And some studies do it on people that move actively and are not padded at all. And so these kind of comparisons are very difficult, which is why I think there's probably something in there where we have to be more careful about when and how we apply these tools.
Yeah, and it's interesting. You said that the motion is observed more with older people and people that have some sort of like, let's say, early dementia, stuff like that. And also, these are the same people that are likely to have reduced gray matter, right? Volume.
Exactly.
So you have like this confounding factor, which is the age or and yeah, I mean, it's that I didn't know about that, and that's a big issue. Like if you can systematically think that let's say old people have a thinning gray matter, I mean that's a known fact, but maybe part of this is due to the added motion, right?
Exactly. Yeah. And also I think there's also some recent work on BMI where you could argue for the same thing, that it's a risk factor for many diseases. also brain-related diseases. But once again, we see this very strong correlations, and it's not quite clear how much of the signal can be explained by motion.
When you did these biases, measure the biases, can you correct other things? Then you can get into really complex sort of statistical model.
Yeah.
Is it just motion or all these people older that move? Then
So for the work that we have in just published in Neuroimage right now, is basically it was more like a proof of concept and validation step. So we had two models. One was just checking the linear correlation, and then we had a linear mixed effects model where we put in age, BMI, sex, motion, and also the cross correlations. And then there was the model that we used to show these effects. Yeah. So as you said, again, it could be arbitrarily complex. And I think for doing more thorough research on, how these biases also affect the brain, the downstream analysis, that would be an interesting follow-up.
yeah, when you show the plots, this is more motion means then the cortex. But if all of the people who move are older, then maybe not the motion you measure and its age.
Yeah, but in the initial plot, these were the same participants. So that here, sorry, it'll take a while.
I mean, if we don't, I understand.
Oh yeah, okay, but yeah. So these are the same people, right, in the same session. Yeah, exactly. But I mean, fair point.
Yeah, and the differences are huge. Like you can go like half a millimeter cube. It's even one. That's like completely different diagnosis. It's based on motion.
25 might be things that is not realistic.
Yeah, exactly. I mean, here, this is the area where we would see this low. Yeah, exactly. This lower score. So these are cases where they would not be excluded from the study. In this case, by the rater that was used in this work.
I have a question for let's say the main problem is that you might have this, let's say you want to have these segmentation tools like FreeSurfer and all these things. They might fail because of different sources, right? You might have all these samples you shown, so you have motion artifacts, you might have tumors that were simulated by the model. And this is only for trying to attack the problem from the motion perspective, right? So do you think there could be like a common factor or common tool to use that for different sources? Because right now, I mean, it's fine. You say, okay, we have this tool, this model, we can put only for motion. Now we're going to do only one for tumors, one for this. So you think something like, for example, the uncertainty repetitions could be useful for these things? Okay, I have a segmentation. Okay, I know for any reason it's very uncertain on these places. So we should or we shouldn't take this into account.
Yeah, so I think it just it depends on the practical approach you have. For example, in the deep learning tools we have now for brain segmentation, they're often using ground truth or corrected ground truth from FreeSurfer. And so FreeSurfer is biased. The bias is in the ground truth, and it's probably possible, but it's not easy to directly say, well, this is an unknown case, or this case is biased, this is not biased, how much? And I think this is why I was arguing for this quantifying first kind of approach, right? Where let's understand first what are the effects, how large are the effects, so on and so forth, to then make more informed decisions on how we have to adjust and what we should do with our toolboxes. And I think in general, I mean, as I said, there's these two big groups, in my opinion, of these confounders, where it's one structural things. And I think there's interesting research going on in this, where you detect out of distribution cases, right? Or you have these uncertainties where you basically say, well, this is not something I've seen in my training data, and then you can react to that, right? And I think that's a easily applicable, very general approach. But probably artifacts you likely have seen in some form or another in your training data. And then I think these methods might not be very applicable, but rather you would have to see specifically, well, what are the biases? What do I expect? How large are they? And so on and so forth. So I think this kind of quantifying and attacking first the understanding, and maybe we can even change something in the scanner, right? Maybe we can just say, well, now that we know, we just fix our position. These kind of things are important, and so that's why now we're focusing more on doing this downstream and not in the tools. But I mean, I hope we can take this to the methods then in the next step, right? But I don't see yet how it can be like solving all the different modes here.
I guess I have a lot of questions. This is more a down-to-earth question. Like when you were showing your motion prediction network, you said you use FCM and you applied it on the scan, right? How do you not run out of GPU memory doing that? First question. And second question is, from my experience, like doing this kind of like volume to either scalar or in your case, you predict over a discrete distribution, it's usually very hard and it requires a lot of training samples because it's just you're going from a massive space to a single. Are you not getting overfit, or like how many training samples do you need to learn that fast?
So now we have 500, and we saw that less basically didn't work well. So we were kind of waiting for more samples to come in, and then okay, we saw the improvement. Now I think we already have 1,000 or 1,500, so I'm excited to try again, retrain and see how well it works now. And yeah, what we saw is that the kind of tricks we did helped with that, right? They were kind of the things. The 3D architecture is the trick is to use only convolutions, right?
Nice.
And then you get less weights.
And you don't run out of memory doing that?
No, so it fits on one GPU, on one A100, I think. And yeah, as I said, this predicting the distribution, that also really seemed to help with regression task, right? To get some kind of more smoothed out gradients, probably, where you can you get more into a good local minimum, I think.
If I understood correctly, for each fMRI, you will assign a motion score for the whole fMRI. But wouldn't it be better if you like assign a score during the time that and assign it for each like time step? Because for example, if it's 10 seconds of fMRI and during the first 5 seconds it's stable, and like the 5 remaining it's moving. I think this way you will have more data to train the model, and it doesn't have to be like 4D for the input. You can use 3D, and this way you can have much more data, and maybe the method will be more stable. And instead of using one single score for the whole fMRI, you can predict the scores for the time and get the average after that. Isn't that much?
I'm not sure if I understand the question 100%. So are you talking about the comparison between methods where we use fMRI as kind of ground truth estimate, or about the learning part where we predict?
Okay. The final goal is that you will assign a motion score for each fMRI.
No, this is not. It's not. So for fMRI, we didn't do the learning for fMRI. We did T1, T2, and FLAIR for the prediction, where we predict from the images the motion. And then for the comparison of the measurements to fMRI, we had one head position where we compared the head position during fMRI to a head position measured by our method, and then check the differences there. So it's a 3D comparison, basically. Yeah, so I mean, it's an interesting thing because there's indeed many things to consider, right? Where you have to average the head position, which is already not trivial, during the fMRI scan. Then you get 2 head positions, one for the fMRI and one for the averaged high frequency signal. And then you compare those and you have to map them to the same reference space. And so, yeah, and that's how we then get this motion trace difference that I talked about. Thank you.
Christian already asked this question, but do you think having a single parameter per acquisition is enough for predicting?
So you mean one score basically per?
So you had, you were estimating right now you're estimating a distribution, but at the end of the day, you're looking for this score. Here's a T1 image, I'm looking at it, and this is my estimated motion. But when that motion happens during the T1, so the T1 acquisition is say 5 minutes on average. So where it happens during that acquisition might lead to very different.
Exactly, for sure.
So you could have the exact same motion in two very different looking images.
Yes, yeah, exactly. So, I think you could make it arbitrarily complex, right? We have like tens of thousands of measurements that are 3D during the acquisition, which I think one of the applications of the camera is also to do online motion correction, right? So you could have, for each measurement, you could have the information of how it was measured and then where the case space was sampled and so on and so forth. And I think it's a very interesting question.
So when you look at the data, did you find any outliers, things where your prediction didn't work, maybe because that was one of the reasons?
Not really, I think. Not always outliers, but I think it's also because we have this data where we don't really see, with our eyes. So but I'm sure, this is a pitfall of the deep learning prediction, for sure, that this relationship between the motion and the resulting quality, that's very abstracted here. And that's why in the work here, one of the applications is just for for understanding the correlations in the population as well, because I think that for that, the average score is very useful, that you just get information of how much people move. But for the image quality, for sure, we need something better in the future. I think the question is just how, again, how many variables do you need, right? How much information do you need, and so that you can still get good learning parameters and get good gradients, and which features do you want to get? And I think it's an interesting learning problem.
Yeah, I have one last one, I guess. I like to think, first, I really wanted to thank as well, beyond the science, but it's the way you present, which is, I find it really cool.
Thank you.
Very simple, very like we get very clearly the idea. That's good. And I like to think a bit outside the box, and I was I was wondering, I really don't remember my physics, but I remember the translation or rotation is in space is something in it's like a shift in the phase in the Fourier space. I think in K space on the reconstruction thing. If you have somebody moving around the scanner, if you think in terms of working in the K space, rotation, so I really forgot my physics, but I remember seeing works exploring these things in fetal imaging, where in the fetus, you have absolutely no control. You can fix a bit the head, but not the fetus. And I remember there was some work exploring these whatever's happening in space is simpler in Fourier space. And I was wondering if this is something that is explored or not at all, or if it could be useful.
So we tried mapping the images back into the K space, into the Fourier space, and then training on the Fourier space images for the prediction. And this did not seem to help. So similar results, a little bit worse. And I think part of this might be explained that we don't actually have the full reconstruction. So we only have the real part of the image, and the imaginary part is actually just deleted. So we will have to acquire data specifically. Ideally, we would just get the raw case-based data, but that's, I don't know, like 100 gigabytes per case or something. It's like super, super big. So there's some logistical problems, but for sure that would be interesting.
And I remember in fetal imaging as well, there were bands in frequencies to capture what is moving and not moving, and there was a lot of more complexity than that. But then somehow it became things much simpler because the motion was easily removed. But yeah, okay.
Yeah,
makes sense.
I think it's maybe still something in there we haven't discovered yet. Maybe with some
because combining those things and then what you're showing here could somehow yes. Okay. All right. Cool. Thanks.
Thank you.
I'll run. I have to teach, so yes.
1 hour.